Hey guys, today we're going to talk about viral replication, so how do viruses actually replicate. If you remember, viruses only attack specific cell types, and that's because of the outer membrane and usually due to the capsid or the glycoprotein composition. So depending on what kind of capsid they have or what kind of glycoproteins, it's going to determine what kind of host cell they have. Okay, so that goes back to host specificity. Okay, so humans really share the same viral diseases with other animals. In the case of like mammals, like all like rabies is like most mammals, but we're not going to usually get something that attacks like a bacteria. Okay, so eukaryotic viruses usually have envelopes on them, and that's because whenever they leave the host cell, they're going to take part of the host cell with it. So therefore, most eukaryotic viruses have some type of cell membrane from the host cell. All right, so there's two main viral replication cycles, the lytic cycle and the lysogenic. So we're going to start out with the lytic cycle. Okay. Um, the lytic cycle, if the name says what it does, it's literally a cycle in which the cell bursts. You remember lytic means slice, okay, to break apart, split. Okay, so this is usually like a super rapid infection and it's usually virulent. Okay, virulent is your new vocabulary word, which means to cause disease or infection. Okay, usually viral virulency is usually talking about how deadly it is and how many people die. So if it's virulent, it's going to cause disease. It's kind of like a pathogen with bacteria, same word. Example of some lytic viruses that you guys have already been exposed to are flu. Okay, Ebola, we've talked about before. COVID-19 is one. SARS, the common cold, rabies, and then tobacco mosaic. Okay, the steps of the lytic cycle include attachment. Okay, the virus has to attach to the cell. Okay, and we're talking about a bacteriophage. Okay, it's going to use its tail fibers. If we're talking about a eukaryotic virus, it's going to be the glycoprotein. Okay, the next thing that has to happen is the virus has to inject its genomic material or its DNA or RNA into the host cell. So the host cell knows what proteins to make. So we call that step penetration or injection. Once again, it just depends on if you're talking about a bacteriophage or a eukaryotic virus. Okay, the next step is replication or biosynthesis. So this is where the viral genetic material, DNA or RNA, is being converted into proteins to make new viruses. So if you remember, viruses only have two components, proteins and nucleic acids. So the nucleic acids are the instructions, so we need to make proteins. So that's when we're going to hijack the host ribosomes and Golgi if we're eukaryotic. Okay, step 4 is the viruses are actually going to assemble and they're going to mature. All the pieces are going to be put together. And then step 5, why it's called a lytic cycle, it's going to reach its burst size in which all the viruses release from the host cell. Okay, and that usually destroys the host cell. Okay, so here's just a little animation that shows you everything I just went over. Okay, so there's an attachment. Okay, there's penetration or injection of the viral DNA. There's replication, assembly, and then there's lysis. Super straight and to the point. Okay, just remember the word lysis means to break open. So literally in the lytic cycle, the virus breaks open. Okay, this is just the same exact kind of information, just in more detail. Okay, so you can read through that when you do your note packets. Okay, this is called the viral growth curve. Okay, it's very, very similar to the bacterial growth curve that we talked about last chapter, except for this little hump right here. Okay, shows penetration. So penetration only takes one virus to penetrate, and then it only takes about 30 minutes for the virus to make its proteins, assemble, and then burst. Okay, so burst time is usually when the curve starts increasing because that's when you're going to release, and then where it plateaus the second time is your burst size. All right, so I wanted you to read this slide on your own, and this is how COVID-19 is virulent. Okay, so what is COVID-19 actually doing to our cells, and why are people becoming sick, and then why are people dying? Okay, because COVID-19 is a eukaryotic virus, which means it's enveloped, but it doesn't destroy the host cell. So this is how you actually die from COVID-19. You can read all those details on your own. Okay, the second type of viral life cycle is the lysogenic cycle, which we call the latent cycle. Okay, so these viruses enter the cell, they still attach, they still penetrate and inject, but they don't go straight into viral replication and making their proteins. They lay dormant. Okay, so the scientific name for dormant is latent. Okay, so they're usually inactive for long periods of time. We're talking up to months or years. Okay, sometimes even decades, depends on the virus. Okay, so what they're doing in the host cell while they're laying low or dormant is they're replicating. They're kind of building a viral army. And then they're going to stay in the host cell until the host is no longer a good host. Then it's going to be activated and it's going to leave, and that's when you see the actual expression of the virus. Okay, so these viruses actually technically use the lysogenic cycle, and then they kick over to the lytic cycle. So we call this the gentle cycle. Okay, so like lysogenic, you still do lytic, but then you got like a little bit of gentle cycle. Examples of lysogenic viruses are HIV, okay, herpes. Okay, if you get herpes, you usually don't have outbreaks all the time. Hep B, and then chickenpox. So what happens in the lysogenic cycle is just like in lytic, the phage or the virus is going to inject its DNA or RNA into the host cell, okay, via attachment. They're going to join the viral DNA with the host DNA. This is new, okay? So as you can see here, okay, when the viral DNA is actually incorporated into the host cell DNA, that's called a prophage. So it's going to maintain its status as a prophage until we get a signal that the host is no longer viable. So they're literally just going to keep doing this cycle over and over. And as the host cell replicates, the virus is actually copying and getting more and more copies of itself. Okay, so a prophage is just when the host DNA and the viral DNA come together. Okay, and every time the host cell replicates, the virus is getting replicated until there's some kind of activation signal. Okay, so the activation signal is when the host is no longer a good host. Okay, so that includes hormone spikes. This is why you see some people have herpes outbreaks during their menstrual cycle. So the spike in hormones, when a girl is ovulating or going to start her menstrual cycle, she'll have a cold sore or genital herpes. Okay, stress is one. Change in diet or exercise routine can have different effects that makes the virus think, oh, this is unstable, I got to get out, and then thus the infection kicks over to lytic. Okay, this is also why shingles appears whenever someone is older, because their body is starting to kind of die and decay. So the virus is trying to get out. And so that's why people have shingles when they're older, not younger. Okay. Once the virus leaves the host DNA, so no longer a prophage, it's literally going to kick over to the lytic cycle and do assembly and maturation and release. Okay. So once again, the virus is said to be virulent because it's going to cause some kind of infection. Okay. Now, some viruses leave the cell intact and they just bud off using the membrane. So that's something like COVID-19 or HIV. Okay, so you can see here, viruses can either go through, okay, the lytic cycle, which we have attachment, penetration, assembly, or once it attaches and penetrates, it can incorporate its DNA into a prophage. It'll just keep replicating, building up its army. It'll get an activation signal and it'll kick back over to lytic. So you can either go around clockwise, or you can go into the lysogenic cycle, divide, activate, and then you're going to kick back over to lytic. Okay, so there's a special type of virus class called retroviruses. Okay, retro means backwards or throw around. So these don't contain DNA, they contain RNA. So they're in the family Retroviridae. So they have an enzyme called reverse transcriptase. Okay, so if you break down that enzyme name, okay, it's an enzyme because it ends in ACE. Transcription is when you go from DNA to RNA, but we're doing the reverse of that. So since this virus has RNA, it's got to convert itself back to DNA. Okay, so you remember the normal genetic flow or central dogma is DNA to RNA to protein. Okay, so the host cell only recognizes DNA. So this virus, whenever it enters, has RNA. So it's got to convert its RNA back to DNA so the host cell can go through transcription translation like normal, thinking it's making its own proteins, but really, in fact, it's building up the virus. Okay, so we call that enzyme reverse transcriptase or RTase. Okay, so it literally just makes RNA into DNA, and then the host cell will carry out normal central dogma. Okay, so two examples of this are HIV, which is the virus that causes AIDS. Okay, and then feline leukemia. So if you've ever had a kitten, the first thing they test is if they have this type of cancer, which is called by the virus. Okay, if you have any questions about tomorrow's quiz, let me know. Okay, I know these videos are short and fast, but you have the note outline. If you need anything, contact me. Thanks.